Hello, I'm Dr. Mark Darcy, and in today's session, I'm basically going to be talking about respiration. Respiration is how we make energy. How we make energy. We use oxygen and we eat food, and then some magic happens in our cells, and we have life. We have energy. We can power the pumps that pump things in and out of our cells and in and out of our intestines. We can build proteins, and we can build bodies. We can dismantle parts of our body, recycle them. We can move muscles. We can do all these things, and respiration allows us to do that. It's a very complicated subject. What I'm going to do in this particular talk is, rather than covering every single detail because there are so many, I'm giving you some highlights. So I'm going to talk about glycolysis, I'm going to talk about the link reaction, I'm going to talk about the Krebs cycle, I'm going to talk about gluconeogenesis, I'm going to talk about ketone bodies and ketosis. And I'm going to give you an overview of the whole lot. There's a lot of biochemistry in this particular talk. I do warn you in advance. The rewind button, the pause button, might be quite useful to understand this particular session. Okay, so without further ado, I'm going to get started. Okay. Okay, so respiration and gluconeogenesis. Now, before I get started on glycolysis, I just want to mention one very important molecule. It's known as ATP, adenosine triphosphate, ATP, and it's the energy currency of the cell. When we eat foods, when we breathe, what we're using a lot of that for is to make something called ATP, adenosine triphosphate. It's a base called adenine, it's a sugar, and it's three phosphates, hence triphosphate. We make this molecule by taking ADP, adenosine diphosphate, and sticking an extra phosphate on it. Now, that phosphate is quite reactive. So when you remove that third phosphate, you turn ATP into ADP and you release energy. That energy can be used to power things. It's a very important molecule. We make ATP, we break it apart, then we stick it back together again. That's why we eat and why we breathe. Another important molecule, which I will talk about a little more as we progress, is known as NAD. NAD is something called a coenzyme. It helps lots of enzymes to basically function properly. It's derived from B vitamins, and it really is responsible for the level of energy to a great degree that we have in our bodies and in our cells. More NAD usually means more energy. Declines sharply with age. A 60-year-old might have half as much NAD in their cells as a 20-year-old, with corresponding lack of energy. But we'll get to that. First stage of respiration, however, is something known as glycolysis. Glycolysis occurs in the cytoplasm of the cell. That's the liquid of your cells. The rest of respiration, or at least a lot of it, occurs in a structure known as the mitochondria. The mitochondria is an organelle. Our bodies have organs, our cells have organelles. The mitochondria is an organelle within our cells that is responsible for huge chunks of respiration, energy production. Okay, so glycolysis occurs in the cytoplasm of the cell, and it's usually the first stage of respiration. The starting compound is one molecule of glucose. The next step is known as phosphorylation. Phosphate is placed on the glucose. We then make something called exos 1,6-bisphosphate. Don't worry about these names, it's not so important, and I will be showing some images very soon. This is just a summary. There's then oxidation of the molecule. There is then oxidation of a molecule known as triose phosphate, which then produces an intermediate molecule. Conversion of the intermediate then follows to pyruvate. Long story short, one molecule of glucose enters glycolysis, and it is broken in half, more or less, to produce two molecules of pyruvate. Glucose is a 6-carbon sugar. Pyruvate is a 3-carbon sugar. One glucose becomes 2 pyruvates. So let's go through the stages. Now, this is a summary. There are many intermediate molecules and enzymes involved, but this is just the bare bones version of respiration. Okay, so we start with glucose, which, as I said, is a 6-carbon sugar. Phosphate is then added to the glucose. The thing that puts the phosphate onto the glucose is ATP, the molecule I just told you about, adenosine triphosphate. It breaks apart, loses a phosphate, becoming ADP, adenosine diphosphate, and the extra phosphate, the third phosphate, is stuck to the glucose, turning glucose into glucose 6-phosphate. There is then what we call an isomerization reaction, which then turns the glucose 6-phosphate into fructose 6-phosphate. When you think about this process, think about a ball sitting on the top of the staircase. It goes nowhere. That's the glucose molecule till you give it a nudge. We use ATP to give it a nudge, and the ball is glucose. When we give it a nudge, it starts falling down those steps and eventually works all the way to the bottom, losing energy as it goes. Now, in the case of a ball, that would be kinetic energy, potential energy turning into kinetic energy. Here we're talking about glucose breaking down and basically producing molecules at the end that are very useful in respiration. So the fructose 6-phosphate, we've just spent an ATP to get the reaction going to make something fairly stable, glucose, into something less stable, glucose 6-phosphate. Now let's break it down. The chain, so glucose, first of all, the most important reason why we phosphorylate our glucose, there's 3 reasons. The first reason is, think about glucose in your blood. You've eaten some food, you've digested the food, there is no sugar in your blood, glucose. It's no use in your blood, it needs to get into your cells. Now, glucose gets into your cells through glucose channels, through glucose transporters specifically. Now, glucose can come in through those transporters into your cells, but the thing is, we want to keep it in the cells. Now, if we didn't do something to keep it in the cells, then what would happen is glucose would enter our cells when we have high blood sugar, but as soon as we end up with more sugar in the cells than outside of the cells, the remainder of the sugar in the blood would not be able to enter the cells because it would be going against its concentration gradient. So the first reason we put phosphate onto glucose is to turn it into another molecule, glucose 6-phosphate, which doesn't fit through those transporters. So it cannot leave the cell again. Plus, this maintains the gradient because normally you would reach equilibrium, same amount of sugar outside, same amount of sugar inside. But there isn't any sugar inside. There is no glucose inside, there's glucose phosphate. Glucose is outside. So it can keep coming in, because as soon as the glucose enters the cell, it's changed into another molecule, so the concentration gradient is maintained. The third reason, and one of the most important reasons that we put a phosphate onto glucose, is to raise it to an higher energy state. Imagine that phosphate kind of hanging off the glucose with a spring, just waiting to be released. It's spring-loaded phosphate. And that starts reactions going. So glucose becomes glucose 6-phosphate, becomes fructose 6-phosphate as that ball bounces down the stairs, and then exose 1,6-bisphosphate. So the exose 1-bisphosphate then is split. So the 6 means we're, oh, by the way, the phosphate, bisphosphate, you can kind of guess what that means. 2 phosphates now. So we spent an ATP here to get the reaction started. Then we spent another ATP here to continue the reaction and allow the molecule to continue breaking down. So we've spent, this is the initial energy investment, we've spent some ATP to get this all going. And here we've got exose 1,6-bisphosphate. It's an exose sugar, 6-carbon sugar, and it's a bisphosphate. There's 2 phosphates on it now, one from this ATP, one from this ATP. So the exose 1,6-bisphosphate then breaks apart. There are enzymes and things involved in all of this. I'll get to some of those later. And you then end up with 2 triose phosphate molecules. So one So we started off with one glucose molecule, one sugar molecule, and now it's become two molecules, two triose phosphates. They are 3 carbon molecules. We've gone from one 6 carbon molecule to 2 3 carbon molecules. There is then an oxidation reaction of these of the triose phosphate. We oxidize it. Oxidation, reduction, oxidation, pulling electrons from something, and the thing that the electrons are pulled from is reduced. Okay, so this triose phosphate, now remember there's 2 of them, 2 times, so this reaction is occurring twice. The triose phosphate then breaks down to an intermediate compound, and as it does so, it generates ATP. It's powering the manufacture of new ATP. So you spent 2 ATPs and you've now got them back. This reaction now, this part of the reaction has produced 2 ATPs. You're now back to where you started. You've got the same number of ATPs that you had when you began. You've also reduced to NADs. So you've reduced to NADs. You've basically turned NAD into NADH. Well, NADH plus, if we're going to be specific. So you've ripped some electrons from the triose phosphate. You've also taken some phosphate from there. That's why it's no longer a phosphate. Phosphate has gone to the ADP to make new ATP. And you've reduced NAD. Now, the reduced NAD is very important. I mentioned earlier that NAD is a coenzyme and helps lots of processes in our bodies work well. It's also very important for the mitochondria, which I will get to later. So at this stage, 2 molecules of ATP are produced by substrate level phosphorylation. A dehydrogenase enzyme, dehydrogenase, it's obviously removing hydrogen, dehydrogenase. The dehydrogenase enzyme removes 2 hydrogen atoms, one from each triose phosphate molecule. The H goes onto the NAD, thus reducing it and making something called NADH. It's happening twice because there's 2 triose phosphate molecules. It gives these hydrogen atoms to NAD, a coenzyme. The triose phosphate has been oxidized as it has lost hydrogen, while the NAD has been reduced as it has gained it. Now let's briefly mention NAD and nicotinamide adenine dinucleotide, NAD. This is NAD, this is NADH, the reduced form. Brain. And here we have nicotinamide adenine dinucleotide, NAD+. So there we can see NAD+. So it's an organic non-protein coenzyme made of 2 linked nucleotides, made in the body from nicotinamide, which is vitamin B3. One of the reasons B vitamins are important. There's 2 riboses in there. Ribose is a sugar, adenine, and 2 phosphate groups. So you can see the phosphates are here. You can also see the sugar here. This is a ribose. There is another ribose. This is the adenine. So NAD, nicotinamide adenine dinucleotide. It's the nicotinamide ring, the ring at the top here, that can accept hydrogen atoms. When NAD accepts 2 hydrogen atoms, it is known as reduced NAD. The reduced NAD transports the hydrogen to the inner mitochondrial membrane, so the NAD is going to the mitochondria to deposit its hydrogens. I'll explain why later. So it will be used to generate more ATP in the electron transport chain. Once it has dropped off the hydrogen, to the mitochondria, the NAD is now back in its oxidized form. It's back in its oxidized form and free to accept more hydrogens. Conversion of the intermediate to pyruvate. So these intermediate compounds then break down to produce pyruvates, generating another 2 ATP. So now we're ahead of the game. We started off with a couple of ATPs that we had to spend. and then we produced a couple to make us even again. Now we've made 2 more ATPs, so we're in profit. A series of enzyme-controlled reactions produce pyruvate, which is a free carbon sugar. Again, 2 molecules of ATP are formed by substrate-level phosphorylation. So here's a summary. We have glucose, which becomes glucose 6-phosphate, then fructose 6-phosphate, exos 1,6-bisphosphate, which produces 2 triose phosphates, free carbon sugars. Sorry, there's 2 of them. Each one is a free carbon sugar. 2 intermediate compounds and 2 pyruvates. So in glycolysis, 2 ATPs are used, but 4 are made, so there's a net profit of 2 ATPs, which can be used for energy. 2 molecules of reduced NAD are sent to the inner membrane of the mitochondrion. 2 molecules of pyruvate are actively transported into the mitochondrial matrix to enter the link reaction under aerobic conditions. Note in anaerobic conditions, the pyruvate remains in the cytoplasm and it's either converted to lactate or ethanol, and lactate can basically be used as an emergency energy source. So we can make lactate in an emergency when we don't have time to involve those mitochondria quite so much, although we can. The lactate is involved in the process actually. I'm sort of missing a bit out, but I'm not going to go into that right now. Lactate, long story short, we can use as an energy source in an emergency. Glycolysis. Glucose 6-carbon. ATPs are spent. We get some intermediate molecules, specifically known as glyceraldehyde 3-phosphate, 3 carbons. These then will generate your NAD. They will reduce it. That is, we've already got the NAD and ATPs. And then we end up with our pyruvates. So 2 pyruvates from 1 glucose. So glycolysis summary: for each glucose molecule entering reaction, 2 molecules of ATP are used, 4 molecules of ATP are produced. So there's a net gain of 2 ATP molecules. 2 molecules of reduced NAD are produced. Glucose is converted to 2 molecules of pyruvate. So let's assume aerobic respiration now, not anaerobic. I'm going to be talking about enough things during this talk as it is, without also covering anaerobic respiration. If O2 is present, free carbon pyruvate passes into the mitochondria. So in the presence of oxygen, the fate of the pyruvate is to go into the mitochondria. If oxygen isn't present, then it stays in the cytoplasm and is used to generate lactate. Here it's in the mitochondria, the pyruvate is completely oxidized, forming carbon dioxide and water. You breathe in oxygen, you breathe out carbon dioxide. Aerobic respiration. The second stage of aerobic respiration is known as the link reaction. So glycolysis is the first stage. The link reaction is the second stage. It links glycolysis in the cytoplasm to the Krebs cycle, also known as the citric acid cycle, in the mitochondria. So it occurs in the mitochondria itself. The link reaction is also in the mitochondria as well as the Krebs cycle. The end product of a link reaction can enter the Krebs cycle. So this is a link reaction. Pyruvate goes in and something called acetyl coenzyme A goes out. Now this is a little bit shorter than glycolysis. There's less steps, nice and easy to go through. So the pyruvate breaks down, and as it breaks down, you reduce some more NAD, meaning you produce some more NADH, some more reduced NAD. You do that by dehydrogenating the pyruvate. There's a dehydrogenase enzyme which basically takes hydrogen off pyruvate, changing its form, and puts the hydrogen onto NAD, thus reducing the NAD. At the same time, a decarboxylase enzyme rips a C and 2 O's, carbon and 2 oxygens from the pyruvate, carbon dioxide. This turns pyruvate into acetate, which is a 2-carbon sugar. You can see where the 3rd carbon has gone. 3 carbons, One goes off here to make carbon dioxide, leaving the acetate as a 2-carbon sugar. Now, the acetate reacts with coenzyme A. Coenzymes help things to do their job. Coenzyme A reacts with the acetate. You now have acetyl coenzyme A. Acetyl coenzyme A then goes to the Krebs cycle, the citric acid cycle. So, summary of the equation: 2 pyruvates plus 2 NADs, NAD plus properly, gives you 2 coenzyme A's. So 2 acetyl coenzyme A's. So we've got 2 pyruvates, 2 NADs, 2 coenzyme A's, gives you 2 reduced NADs, 2 acetyl coenzyme A's, and 2 carbon dioxides. So coenzyme A, made from another B group vitamin, adenosine, which is ribose, and adenine, sugar and a base. And then there are 3 phosphate groups and a cysteine. The purpose of the coenzyme A, it carries the acetate, known as ethanol-8, groups made in the link reaction into the Krebs cycle. So to sort of summarize it here, pyruvate goes in, acetyl coenzyme A goes out. And in that process, NAD is reduced, thus oxidizing the pyruvate. The pyruvate is decarboxylated, meaning CO2 leaves there, and coenzyme A binds to it, giving acetyl coenzyme A. So 1 carbon atom is removed from pyruvate in the form of CO2. The remaining 2 carbon molecule combines with coenzyme A to produce acetyl coenzyme A, the acetyl in the coenzyme A. Another oxidation reaction occurs when NAD plus collects more hydrogen ions. This forms reduced NAD. No ATP is produced in this reaction, so you don't get any ATP generated in the link reaction. Obviously, the link reaction occurs twice because we have 2 pyruvates. So all those numbers of NADs we've just talked about, double it. For each glucose molecule used in glycolysis, 2 pyruvate molecules are made. But the link reaction uses only 1 pyruvate molecule, so the link reaction of the Krebs cycle happens twice for every glucose molecule that goes through glycolysis. So if we look here, we can see the overall equation for one link reaction and Krebs cycle. Pyruvate plus NAD plus coenzyme A gives you acetyl coenzyme A plus reduced NAD plus CO2. The products of the link reaction go to the Krebs cycle and the electron transport chain. So for each glucose molecule, 2 acetyl coenzyme A go into the Krebs cycle, 2 carbon dioxide are released as waste products, and 2 reduced NAD go to the electron transport chain of the mitochondria. Krebs cycle. Named after Sir Hans Krebs in 1937. He got a Nobel Prize for it. He discovered this entire cycle, which, as you'll see shortly, is quite complicated, and he did it all in an old-fashioned lab over in Oxford. You imagine it, lots of bubbling tubes and beakers and Bunsen burners and all of that sort of thing going on. No computers, no fancy machines, no lasers, yet he still managed to work out this very complex and very small cycle within the cells. They got a Nobel Prize for it as a result. I should say that it was more his technicians that did the graft of the work. He was more of a theoretical scientist. Now, my PhD supervisor, this is a long connection, I admit, but my PhD supervisor had his own PhD supervisor back in the 80s, and his PhD supervisor was Hans Krebs. So there is a link there. Hans Krebs, so there's a story I've got for you, basically, which has been handed down a couple of levels. Something might have got lost in translation, but it's a good story anyway. And it goes like this. Everyone in the lab knew that Hans Krebs was terrible at practical work. He was good at ideas, he was terrible in the lab. His technicians did the work. One day, he walked into the lab, and they were making, I can't remember what it was, but they were making something flammable. in a big beaker. They were making something quite flammable, and it was in this huge beaker on a tripod. Now, this is the 30s, so not the health and safety we have now. This is over at Oxford University, by the way. So Hans Krebs walks in with a cigarette in his mouth, and a spark comes out and lands in the gas just above the liquid, and the gas started to flame. Now, when that gas burned out, that flame was going to go down into the liquid, and the whole beaker was going to explode, blowing up the lab. Luckily, there was a quick-thinking lab technician in the room. He grabbed the beaker full of liquid with the flame at the top, went to the nearest window and threw it outside into the courtyard, and it exploded. Luckily, no one was walking by. So otherwise, it would have been an entirely different situation. But that shows you that Hans Krebs was not particularly good in the lab, even if he was a good ideas man. Anyway, it's also known as the citric acid cycle or the tricarboxylic acid cycle, TCA cycle. It takes place in the matrix of the mitochondria, the matrix of the mitochondria. That's like the cytoplasm of the mitochondria. From one glucose molecule, you get 2 pyruvate molecules. That's glycolysis. So the Krebs cycle goes around twice per glucose molecule. And this is a summary of the Krebs cycle. It's a circle. Moves all the way around, and in the process, reduces NAD, makes ATP, reduces something called FAD, and reduces some more NAD. So we make a lot of reduced NAD in this cycle, and we also produce a little bit of ATP. ATP used for energy. NAD is going to be used in the electron transport chain of the mitochondria to generate lots of ATP, lots and lots of ATP. Now, it's hard to remember this, just looking at it, not that you necessarily want to, but if you do want to, there is a trick. There is a trick. Sounds silly, but it works. Dena, dena, afana. Dena, dena, afana. Dena, dena, afana. It's a tune. So, respiration is all about releasing energy from food. Oxidation releases energy. When a carbon compound is oxidized, coenzymes are reduced. The coenzymes involved are NAD and FAD. Decarboxylation is removal of CO2. And remember this, 6, 6, 5 and 5 4s. Well, I don't need to remember that really, but see what I mean. Okay, so, dena dena afana. Decarboxylation and production of reduced NAD. That's DINA, decarboxylation, production of reduced NAD. DENA, decarboxylation and production of reduced NAD. A, production of ATP. FA, production of reduced FAD. Na, production of reduced NAD. So, DINA, DINA, AFANA, DINA, decarboxylation and production of reduced NAD. DENA, decarboxylation and production of reduced NAD. There's the CO2, decarboxylation, production of reduced NAD. A, production of ATP. FA, production of reduced FAD. Na, production of reduced NAD, but in this case, without decarboxylation, no carbon. So, DENA, DENA, A, FA, Na. That's how you remember it. That's how you remember it. Go through it a few times and it will stick. Dina, dina, afana. Moving on. Krebs, honestly, I promise, it does stick when you say it a few times. And look at the Krebs cycle while you're doing it. So Krebs cycle summary. Each acetyl coenzyme A entering the cycle results in 2 carbon dioxide molecules, 1 ATP molecule, 4 pairs of hydrogen atoms, later used in the electron transport chain to produce ATP. Used to reduce NAD and FAD. 3 reduced NAD are produced and 1 reduced FAD per cycle. NAD, nicotinamide, adenine dinucleotide, FAD, flavine, adenine dinucleotide. So link and Krebs cycle summary. Both take place in the mitochondrial matrix, liquid of the cytoplasm of the mitochondria, series of enzyme-controlled reactions. Remember, 1 glucose molecule produces 2 pyruvate molecules during glycolysis. So, here are the numbers that we get. I should leave that up for a second. So you can see what we get from the link reaction and what we get from the Krebs cycle. Okay, so there's the link reaction feeding into the Krebs cycle. And here's a summary of the whole thing so far. So far, we're not done yet. There's glycolysis in the cytoplasm, then within the mitochondria, the link reaction that then feeds into the Krebs cycle. Next, we have this here, the electron transport chain, which we should move on with now. So first a summary, one final summary. Anaerobic respiration, don't really need to know this, I didn't really cover it, but anaerobic respiration makes 2 ATPs per glucose. That's basically it. It's just glycolysis. That's all you're getting, those 2 ATPs. Aerobic respiration makes 32 ATP per glucose. If you've counted the ATPs we produced in glycolysis and the Krebs cycle, we've not got to 32, have we? So there's a few more to go. Anaerobic respiration only completes glycolysis, which makes 2 ATP. Hence, this is why anaerobic respiration only makes 2 ATPs per glucose. Aerobic respiration makes 32 ATPs because 2 ATPs come from glycolysis, 2 from Krebs cycle, as it happens twice, and then we get the rest from the electron transport chain. So where does it occur? It occurs in the inner mitochondrial membrane. So mitochondria has an outer membrane and an inner membrane. And the inner membrane is lined with a bunch of enzymes which perform these processes. They're known as the electron transport chain. Okay, so the membrane contains electron carriers. They're embedded. They're like protein complexes. Well, they are protein complexes. The folded cristae of this membrane increases the surface area for housing the protein complexes. Folded cristae just means the inner membrane is folded. Little rivulets, like a river zigzagging. Results in the net synthesis of ATP and water. It's a series of coupled redox reactions. That means reduction, oxidation, reduction, oxidation. One molecule is reduced, another is oxidized. Again. So coupled redox reactions catalyzed by oxidoreductase enzymes and requires oxygen as the final electron acceptor for the process to work. That'll make more sense when I get to it. Okay, let's get started. So what we're seeing here, we're seeing these protein complexes. These line the inner mitochondrial membrane, the cristae. So out here would be the cytoplasm. In here would be the matrix of the mitochondria, and here are the enzyme, the respiratory enzymes. This here is something called ATP synthase. This is where we actually make the ATP. We'll get to that shortly. Okay, so these are the complexes. I'm not going to go through the naming of them all, but that's what they are. Complex 1, 2, 3, 4, 5. And this is what they do. Okay, so let's get started. You might want to focus on this bit. 1. Reduced NAD. Remember, we produced a lot of reduced NAD. We produced a lot of reduced NAD so far from glycolysis and from the Krebs cycle. Reduced NAD is reduced because it's got an H on it. Dehydrogenases pulled hydrogen off these sugars, added them to NAD, making it NADH, which is reduced NAD. So reduced NAD delivers 2 hydrogen atoms to complex 1. So here we go. Reduced NAD delivers 2 hydrogens to complex 1. Drops them off, turning it back into NAD. It's now oxidized again. Well, no longer reduced. And this NAD can then go back to glycolysis and all the other processes and do its thing. So reduced NAD delivers 2 hydrogen atoms to complex 1. These atoms split into 2 H+ ions and 2 electrons. So what does that mean? Well, a hydrogen just has 1 proton in the middle, positively charged, one electron in orbit, negatively charged. So if you split that hydrogen up, you're basically splitting it into protons and electrons. We call these protons H plus ions, and electrons we just say E minus. So reduced NAD delivers 2 hydrogen atoms to complex 1. These atoms split into 2 H plus ions and 2 electrons. The electrons are passed on to complex 2. So here they are, and there they go to complex 2. So we've had a reduction-oxidation reaction here. Okay, they're passed on to complex 2, and this process releases enough energy to pump the H plus, the protons, against the concentration gradient from the matrix into the intermembrane space of the mitochondria. So from the cytoplasm into that space between the inner mitochondria membrane and the outer mitochondrial membrane. Why do we do that? Why do we do that? Why do we want it to be in the space? Well, the thing is, we actually want the mitochondria to be positively charged inside. Dropping those protons off makes it positively charged inside compared with outside the mitochondria, which is negatively charged, which is neutral. And as a result, you've got this motive force building up. The protons want to leave, but they can't. They will be allowed to leave eventually through this channel, ATP synthase. Now, ATP synthase, imagine it like a water wheel. As those protons, imagine them, they're water going down a waterfall. As they hit this enzyme, they rotate the blades, but instead of generating energy the normal way that water would, what we're doing is we're using that movement to take ADP, adenosine diphosphate, to stick it with a phosphate to make ATP. That's what ATP synthase does. But we'll get to that. So, step 1, we basically add reduced NAD, come to complex 1. It's been split into H plus ions and electrons. The electrons have moved to complex 2, and the protons have pumped into the intermembrane space, making it positively charged. 2. The electrons from NAD continue from complex 2 to complex 3. The energy released at this stage is not enough to pump H plus ions from the matrix into the intermembrane space. Reduced FAD from the Krebs cycle drops off 2 H atoms directly to complex 2, bypassing complex 1. So there's the FAD going straight to complex 2, and therefore dropping off its 2 H atoms. Again, these hydrogen atoms split into 2 H pluses and 2 electrons. The electrons are passed on to complex 3, but the H plus ions remain in the matrix due to lack of energy for pumping at this stage. The 4 electrons, the 2 from the NAD and the 2 from the FAD, pass from complex 3, there they both are, to complex 4. The energy released is enough to pump H plus ions from the matrix into the intermembrane space. The 4 electrons move from complex 4 into the matrix, where they combine with 4 H plus ions and O2 to form H2O, to form water. The energy released at this stage is enough to pump H plus ions into the intermembrane space. Then we have chemiosmosis. The pumping of H plus ions into the intermembrane space generates an electrochemical gradient and a pH gradient. Basically, lots of protons inside, none outside, they want to leave. So this is known as a proton motive force that should lead, in theory, to a diffusion of H plus ions back into the matrix. However, the membrane is relatively impermeable to H plus ions, and the only way they can return to the matrix is through the ATP synthase enzyme complex, the water wheel. As the H plus moves through this complex, it catalyzes the formation of ATP from ADP and phosphate, sticks them together. That's what the energy is used for, to make ATP. It is this stage that is known as oxidative phosphorylation. You are phosphorylating the ADP, putting an extra phosphate on it to form ATP by using oxygen as the final electron acceptor of the electron transport chain, which is why without oxygen, you die. So, how much ATP do we get from a molecule of glucose? Each molecule of reduced NAD contributes 3 ATP molecules in the electron transport chain. As reduced FAD bypasses complex 1, each reduced FAD molecule only contributes 2 ATPs to the electron transport chain. So we get 38. But not really. It's not an efficient process. Some of the ATP produced is used to actively transport, to move, the pyruvate from the cytoplasm of a cell to the matrix of the mitochondria. It doesn't go there by itself. Energy is used to transport it. Some ATP is used to bring reduced NAD from glycolysis in the cytoplasm into mitochondria. Some energy is used to transport ADP from the cytoplasm to the mitochondria. Plus, the membranes aren't completely impermeable. Some H+ ions leak back across the mitochondrial membrane, reducing the proton motive force that generates ATP. So, in reality, it's more like this. Each reduced NAD actually contributes about 2.5 ATP rather than 3. Each reduced FAD actually contributes about 1.5 ATP rather than 2. So about 32. So not 38, 32. Remember that. So details: in complex 4, the electrons are combined with protons and molecular oxygen to form water. The oxygen diffuses in from the tissue fluid. Oxygen is only involved at the very last stage of respiration as the final electron acceptor. The energy of the electrons is now stored in the form of a proton gradient across the inner mitochondrial membrane. The ATP synthase enzyme has a proton channel through it, and as the protons fall down this channel, the energy is used to make ATP. It takes 3 protons to synthesize 1 ATP molecule. This method of storing energy by creating a proton gradient across a membrane is called chemiosmosis. Get that in your head. You might want to take a pause now, because we're about to move away from pure respiration. So we talked about glycolysis, the link reaction, the Krebs cycle, and the electron transport chain. We're now going to cover a slightly different topic. It all links together, but if your head is already a bit numb now, you might want to pause for a while before moving on. I'm going to move on right now. Okay, gluconeogenesis. What is it? It's a synthesis of glucose from non-carbohydrate sources. So we store our glucose. When we eat, the glucose goes into our bloodstream. Some of it ends up in our cells, but the extra stuff is stored as glycogen. You stick lots of glucoses together, you get glycogen, you store it in the liver. You've got about 2,500 calories of the stuff in your liver. And you break it down, turn it back into sugar when you need it. I'm not going to talk about that process. It's basically just, it's just hydrolysis. It's just hydrolysis. You add water, basically, to break them apart. But there's other ways of getting carbohydrates if glucose isn't available. It's important during exercise and starvation. During exercise, needs for glucose can outcompete the supply. We can't break down that glycogen quick enough. We just need our energy quickly. Also, if we starve ourselves for just a couple of days, then we use all of the glycogen stores from our liver up. So we have no glucose in our body, meaning we need to make some or use an alternative energy source. I'll talk about the alternative energy source at the end of the lecture. So it's not the reverse of glycolysis. The substrates, glycolysis, by the way, glycolysis, we've gone through. It's not just the reverse of that to get glucose. It's a bit more complicated. The substrates are pyruvate and lactate. We'll move on. Not made from fatty acids. Those are ketone bodies. I'll talk about those later. Takes place in the liver. Takes place in the liver. That's where we're making our glucose. So at least 90% of it does. 10% in the kidneys. Uses ATP. And you don't need to memorize this. There's a lot of chemical reactions. Now here I'm including some of the intermediate molecules that I didn't talk about earlier. Shows you that these processes are more complicated than you may have initially seen. There's the glyceraldehyde, you recognize that one there. Here's the pyruvate, you recognize that one there. There's a few intermediates that we didn't go through, and I'm not going to go through in a lot of detail right now. I just want you to be aware that there's a lot of sub-steps to these steps. Here was where we produced the reduced NAD. Here's some ATP. Here's some more ATP. Pyruvate kinase is just the enzyme that allows for the formation of the pyruvate. Okay. So that's sort of a summary of glycolysis in a bit more detail than you maybe had earlier. Well, a summary of the end part of it anyway. This bit's here, this particular stage is irreversible. You can see these two stages can be reversed. This reaction can go either way. So can this reaction. So can this reaction. This is irreversible, an irreversible reaction. This phosphoenol pyruvate to pyruvate, that's irreversible, which is why the biochemistry gets so complicated. So, lactates, amino acids, these basically lactates and amino acids, they can be used. They can be used as an energy source. We can use protein as an energy source. We can use protein. We can use fats as an energy source. So we can make lactates. We can use amino acids. We can use them as an energy source. And we can basically, let's go through this. So basically, we can produce pyruvate from them. And then we get ATP being spent, producing ADP. That produces oxaloacetate. Amino acids can actually go directly into this step. So here is where we can enter this process, and also here we can enter this pathway. Oh, whoops, wrong way. And this reaction here can go both ways. We can convert this oxaloacetate into phosphoenol pyruvate. Step 1, pyruvate to oxaloacetate. Something wrong with my voice today. The enzyme involved is pyruvate carboxylase. Carboxylase, remember decarboxylases, remove carbons. The carboxylase is going to put one back. If we're going to get to glucose, we have to add some carbons, don't we? And some hydrogens for that matter. Uses ATP. Takes place in the mitochondria. Then we move to the cytosol. So oxaloacetate, we spend some NAD, so to speak. We take NADH, convert it to NAD. So we're basically putting hydrogens back, as we did previously, and we get malate. We get malate. As we can see, we have these two H's here. Here we have an extra two H's. That's what you get from oxaloacetate to malate, which is now going to go into the cytosol, leaving the mitochondria. So now the malate, there's going to be another, there's going to be an NAD here, which is going to produce NADH and go back into oxaloacetate. You might wonder, why did we turn the oxaloacetate to malate, take the malate out and then convert it right back to oxaloacetate? Well, because the oxaloacetate can't get through that membrane on its own in its current form. The enzyme is malate dehydrogenase. Step 2, the oxaloacetate plus a molecule called GTP, ATP, adenosine triphosphate, GTP, guanosine triphosphate. Guanine is another base. ATGC. They're all bases in our DNA, but they can also be useful outside of our DNA. Adenine with phosphates and sugar gives you ATP. Guanine with phosphates and sugar gives you GTP. Anyway, we've gone from oxaloacetate now to phosphoenol pyruvate plus a couple of GDP. Gone from GTP to GDP. Phosphate has been released. Triphosphate to diphosphate. And there is the phosphate right here. Plus CO2. The enzyme here is phosphoenol pyruvate carboxykinase. Uses GTP. Takes place in the cytosol of the cell. So all reversible reactions. So when we look here, we can see each of these reactions here are reversible. So we can basically move up this chain until we get to glyceraldehyde. Remember glyceraldehyde from earlier? A couple of phosphates are lost, dephosphorylation, so the bisphosphate is losing one of its phosphates, another, no longer, so now we're losing phosphates, we're going to glyceraldehyde, free phosphate. Glyceraldehyde, free phosphate. This again, this reaction can move both ways, so to dihydroxyacetone phosphate. So glycerol can enter the process here. Glycerol enters the pathway. Let me just go back there. So glycerol enters the pathway via the dihydroxyacetone. Dihydroxyacetone, DAP, DAP. So glycerol can bind in there and enter the pathway via the DAP. So glycerol, there's some ATP being spent to make ADP. We now get glycerol phosphate. Glycerol phosphate dehydrogenase gets involved. So here glycerol kinase is the enzyme that causes this reaction to take place. Here glycerol phosphate dehydrogenase causes this reaction to take place. Dehydrogenase. And then here at the end, we have dihydroxyacetone phosphate. So that's how we get the dihydroxyacetone phosphate. This is how glycerol enters the equation. Glycerol phosphate intermediate, that's what we've got. Uses ATP but generates NADH. So glycerol comes along, we get the dihydroxyacetone phosphate, then glyceraldehyde free phosphate. Aldolase is the enzyme that again, this is a reaction that can go both ways. This produces fructose 1,6-bisphosphate. You can see how we're moving up that glycolytic chain. Remember fructose? We're pretty close to glucose now, pretty close to glucose. So here we go, the fructose 1,6-bisphosphate, ADP, ATP, gives us fructose 6-phosphate. These are the enzymes that cause this to occur. So phosphofructokinase, then phosphoglucose isomerase gives us glucose 6-phosphate. We're one step away now. We're back to the phosphorylated glucose. This phosphorylated glucose, glucose 6-phosphate. Glycogen can actually produce this as well. If we break down glycogen, if we've got any in our liver, we go directly to this step here, the glucose 6-phosphate. So this bit, irreversible. So, well, yes, so there's glycerol, dihydroxyacetone phosphate, glyceraldehyde free phosphate, fructose 6-bisphosphate, fructose 6-phosphate, glucose 6-phosphate. I think I'm showing you the same slide twice here. Sorry about that. I think I've duplicated a slide or two. I have gone through this. No, I didn't go through the top part. Okay, there we go. This bit at the top here is irreversible. The glucose down to glucose 6-phosphate. So here we go all the way to glucose 6-phosphate. And then from glucose 6-phosphate to glucose, we use an enzyme called glucose 6-phosphatase. Just removes the phosphate, that's all. You remove the phosphate from glucose 6-phosphate, you get glucose. We've made glucose. We've made glucose from amino acids, a lactate. We've made this glucose, and this glucose, this glucose can be used as an energy source. And cell's complicated. There's lots and lots of biochemistry. So final steps of gluconeogenesis takes place in the endoplasmic reticulum of the liver and kidney. Remember, livers and kidneys. Within the livers and the kidneys, we have lots of endoplasmic reticulum. Endoplasmic reticulum are just these structures. All our cells have them, actually. They are the sites of all sorts of little intricate processes, including gluconeogenesis. So this is the membrane of endoplasmic reticulum. So imagine these endoplasmic reticulum floating around in the cells. They have a membrane. In that membrane, there is the enzyme glucose 6-phosphatase, which turns the glucose 6-phosphate into glucose by removing a phosphate. We've made glucose. This glucose now can go out through this T3 channel into the cell itself, out of the endoplasmic reticulum and into the cell itself. It can be used as energy, or when the liver and kidneys are producing it, it can be used for all sorts of processes in the body. So this needs to be regulated. And we mentioned a lot of those enzymes. These enzymes can be up or down regulated. There's all sorts of different processes that can regulate this. I'll let you go through that in your own time, but I will summarize. It's linked to glycolysis, activated by something called citrate and acetyl coenzyme A, inhibited by AMP, that's adenosine monophosphate, and ADP. Also fructose 2,6-bisphosphate in the liver. So summary of gluconeogenesis. I'm sure this is all stuck in your head first time, but if not, here's a summary. Glucose to glucose 6-phosphate can go both ways. Glucose 6-phosphate to fructose 6-phosphate, both ways. Fructose 6-phosphate, then we can go fructose 1,6-bisphosphate. This is where the dihydroxyacetone phosphate comes in if you have glycerol, and the glyceraldehyde 3-phosphate. Then here, several steps. Glyceraldehyde 3-phosphate to phosphophenols can go again both ways. Oxaloacetate can go into phosphophenols. Pyruvate can go into oxaloacetate. These are amino acids. So these are amino acids. Proteins, or we're using proteins here as an energy source. Amino acids can go directly to the pyruvate pathway as well. We can also use alcohol. So pyruvate, can be broken down into alcohol in anaerobic respiration. Can be broken down into acetyl coenzyme A, which I'm going to talk about in my last couple of slides. And lactate. Lactate can be used as an energy source. Acetyl coenzyme A also. We'll get to that. Okay, so that's a summary of this particular pathway. And there's the glycogen at the top. So I'm going to finish with a brief few words about ketone bodies. What are ketone bodies? You've probably heard about ketogenic diets and ketosis and all of these things. Diabetics do their best to not go into ketosis. Very high ketone levels in your body can cause something called ketoacidosis, which can damage your cells. It can be very harmful. But mild ketosis, a lot of recent research has shown, is actually quite good for you. We're not designed just to run on glucose. We aren't. We live on glucose now because carbs are easy to get a hold of. We all eat rice, we eat potatoes, we eat fruit, we eat pasta. Most of us have a diet that's very high in carbohydrates, so our body pretty much uses glucose, which means glycolysis. But we don't have to use glucose. When we run out of glucose, we can in an emergency make glucose in the pathway I've just told you about, gluconeogenesis, but we don't have to use glucose at all. And if you go back to ancient times, back into the Paleolithic times, humans wouldn't have that much carb in their diet. We were hunter-gatherers. We would eat meat. We would maybe get a few tubers out of the ground. We wouldn't get a lot of carb. We might get a lot of fat. We might get a lot of protein. And we've just explained how we can use protein to make glucose, but we can actually also use fat to make glucose. Imagine some ancient caveman. Lots of food during the summer, not so much in the winter. He spends the summer building up fat reserves. Any extra glucose that you get in your diet, you can convert into fat and store it. In winter, no more food around. You start burning up the fat as an energy. You'll burn protein first. So once you run out of carbs, you'll start burning protein, but your body won't do that for very long. It doesn't like to use amino acids as an energy source, because if you use protein for more than a brief period of time, well, there's not many sources of it in your body, and one of the major sources are your muscles. So you start eating your own muscles for energy. So you don't tend to stay in that state. So rapidly you switch from using amino acids to using fat. And long story short, you turn fat into these things, these molecules called ketones, ketone bodies. Well, ketones. Let's just say ketones. Ketones include, well, there's many ketones, but one of the most important is beta-hydroxybutyrate. So excess acetyl coenzyme A gets released on the oxidation of fatty acids. So when you oxidize your fatty acids, you basically produce acetyl coenzyme A, and if there's a surplus of it, or if there's a need for it, and it's not required immediately by the liver, it gets converted into ketone bodies. Acetoacetate is one, and D-beta-hydroxybutyrate is another. They are circulated by the blood to all the peripheral tissues of the body to be used as fuel for the citric acid cycle, completely bypassing glycolysis, going right past it, going straight to mitochondria. When you use ketone bodies as an energy source, no glycolysis. It's an emergency, and it works very well. The ketone bodies are a transport form of acetyl groups, basically. It's a way of getting acetyl around your body to your cells. They can supply a large fraction of the energy of some peripheral tissues, up to 1/3 in the case of the heart. And people that go on to ketogenic diets, pretty much all of their energy is from fat. If you cut out the carbs from your diet, your body will switch after a few days to 2 weeks to burning just fats. And you can eat a diet high in fat with no carbs, and you don't gain weight because the fat is being used as an energy source. This is very good for people that have basically become a bit too... Okay, so benefits of ketogenic diet, benefits of using fat as an energy source. Now, when you use carbs as an energy source, to basically get the carbs out of your blood, you need to produce insulin. You need to produce insulin. And insulin production, overproduction, can lead to diabetes, which is bad. Loss of insulin sensitivity. And that's bad and can cause all sorts of conditions. Diabetes is an awful condition, can cause a much shortened life. You don't produce the insulin because you're using something else as an energy source, the ketones, then you don't get half of the problems that you do when you're living on a primarily carb diet. Also, insulin triggers a pathway in your cells known as the TOR pathway. Basically, it stimulates something called IGF, insulin-like growth factor, which basically causes cells to get into a kind of grow and divide state, which can lead to cancer. Also, inflammation. Too much insulin leads to inflammation. And so many diseases, especially diseases of old age, are caused by inflammation, from arthritis, autoimmune diseases, Alzheimer's. All of these things can be caused by, well, to a degree, by producing too much insulin. If you live on a fat diet rather than a carb diet, and you use ketones as an energy source, then you're not really producing insulin. And this has been shown to have huge health benefits. Okay, ketone bodies, types: acetoacetate, beta-hydroxybutyrate, acetone. The location and of the synthesis is in the liver mitochondria, primarily. Location of utilization, mitochondria of all peripheral tissues, including the brain. The brain can live on glucose. You can also use beta-hydroxybutyrate as an energy source. Reasons for importance: they are water soluble, need no carriers in blood. They are produced in the liver when the amount of acetyl coenzyme A present exceeds its oxidative capacity. Used by extra hepatic tissues, non-liver tissues, in proportion to their blood level. Okay, I'll probably do another lecture which will be all about ketogenesis, ketosis, and ketone diets. But this one, I think I've covered enough for now. Thank you very much for watching. I look forward to speaking with you all again very, very soon. Okay, thanks again for listening, and have a great day. Goodbye.